So today I'm going to talk to you about the powdery mildew of the grapevine and also botrytis bunch rot and summer bunch rot complex, which I'm trying to give you a little bit more information to distinguish between what is the summer bunch rot and botrytis bunch rot is. And we have been having this problem. Anyway, so I want to start with botrytis. Powdery mildew one. So this is my email address. If you would like to contact me and then need more information, I will be happy to give you that information as well. Little bit outline of my talk. I'm going to give you a little bit introduction, talk about powdery mildew, summer bunch rot, and my lab has been running field fungicide efficacy trials for multiple years. I'm just going to share some of those information as well, and then leave you with a conclusion. So a little bit more background information about the powdery mildew. So the scientific name of this disease is Erysiphil nicator, formerly known as Uncinilla nicator. So both names are the correct. But the most common name is right now is the Erysiphil nicator. It is a fungal pathogen. It is an obligate. That means it requires living organism to survive. It cannot survive without any living organism. However, it also produces some overwintering structure where they can also do the overwintering, which I'm gonna give you a little bit more information. So this disease is native to North America. It was first described in 1834. So which means that this pathogen is well adapted to our condition, where it can survive and then cause more damage on grapevine. After its introduction to Europe, it was introduction rest of the world, including California in 1859. We still don't know the exact date, but those are the times that a pathogen was identified, and later on in Australia in 1866. So I'm just going to give you a little bit a biology of or infection mode of the pathogen, so you can have a better understanding what's happening from the beginning of the season in your vineyard. So imagine this is a surface of grapevine leaves. When the single spores, that could be probably at the beginning of the season, ascospores, which is a sexual fruiting body, that I'm going to show you a little bit more information about that, can land on the surface of the leaves at the early beginning of the season, when the shoots are certain time, when the temperature comes in certain time. So that single spores can germinate like a seed, and then they produce a structure called aprestorium, which means that when the pathogen is trying to penetrate into the tissue, it is trying to produce more mycelium in that area, so it can move into the tissue forcefully. Or it can also penetrate into the plant and tissue through the natural opening, which is on the leaves, could be stromata. Once they get into the tissue, and then from that on, they can start utilizing the nutrition and destroying that part of the tissue. And then when they are done at that part of the life, they start producing more conidia, which is asexual fruiting bodies. These asexual fruiting bodies are identical to one another. So that requires, or that keep the same genetic material, okay? And this life cycle seems to be long-term, but within the 6 to 8 days that completed that life cycle, okay? So that cycle, if the conditions are good, they complete and complete. That means once you have the infection, once you cannot control or prevent, that's going to continue having the infection. Because of this, preventative management of the powdery mildew is very, very important. These are the common symptoms. I'm sure you all are familiar with how to identify, what to look for, but by the time when you start seeing these symptoms on the leaves, it is too late to control. That means you have already a lot of spores are flying around in your vineyard under the canopy. So these are the first kind of symptoms that you can see powdery on the fruit, and this is the fruit that is completely covered with the pathogen. In late season, when the pathogen is not controlled and then colonized on the surface of the berries, of course, they kill the tissues on the surface berry part. When plant or pathogen, the cluster continue expanding it, once those tissues are dead, they can start cracking it. So you will see a lot of cracked berries on the heavily infestation areas. So this is the too late season. You don't want to have these kind of things in your vineyard. Again, the pathogen also could colonize on the green tissues, as you can see here, and that green tissue can turn into just before the winter, it can just get into, and then these kind of symptoms are very, very common and common. As I mentioned you, that the pathogen produces two kinds of spores. One of them is the conidia, which is asexual fruiting bodies. It can also produce cosmothecia, which is a sexual fruiting bodies that requires mating type A and B. That's how we determine the sexual, the part of the pathogens. And then these are the very heavily infested the berries that you can start seeing these brown and black spots. Those are the cosmothecia. So the brown ones are the immature part of the cosmothecia. That means the pathogen is continue producing those fruiting bodies. These are the very characteristic, the shape of the cosmothecia. So these are the asa, which is a part of the fruiting bodies where a pathogen produce 8 ascospores. These ascospores can stay in the bodies during the winter, whenever we had the small amount of the precipitation. That could be rain, that could be also the dew point in the vineyard. They continue releasing those spores. But most common, one important one is that the early springtime, when the temperature gets into the certain time where pathogen can utilize colonization on the healthy plants. In the past, my predecessor, late Doug Gubler, has done some research, and they also identified some of those, the mycelium of the powdery mildew pathogen in dormant buds. So these are a few pictures that shows how they survive in those dormant buds. But again, the amount of this survival is very low. Majority of the infestation comes from the overwintering structure cosmothecia in the field. What makes a bad disease year? I'm sure you can answer this question by now, because last year our conditions were so perfect for the disease to flourish, and that was a bad year. So in the plant pathology discipline, the way that we describe this is that we call it a disease triangle. In disease triangle, host is the grapevine, pathogen is the powdery mildew pathogen erysipeniculator. Without the right environmental condition, you are not going to have the disease. Okay, so it doesn't matter what the conditions are, you have to have 3 conditions together in order to have the disease. Because of that, if you cannot change the host and pathogen, management of the environmental condition can give you advantages, which over the talk, I'm going to talk to you and give you a lot more information about that. So in this point, I would like to talk to you a little bit advantage of the sulfur. Sulfur is come up with a couple of the form. One of them is the lime sulfur. Lime sulfur is the only product that could be applied during the dormant season. According to Gubler research, this could target Cosmothecia during the dormant season. Again, this lime sulfur cannot be applied during the growing season. It can burn leaves. So that's one alternative that could be applied. And it also comes with the dusting and sulfur and wettable sulfur, different form. I'm sure you all know. So they all are the form of the coming. So what is the advantage of the sulfur? It's a natural product. It is inexpensive and effective. It has been used more than 100 years, and then there is no case of the resistance. The reason for that, sulfur has two kinds of mode of action. One of the mode of action is that it has a direct effect when it get contact with the spores, ascospores, or other part of the pathogen that kills. And also, when the temperature gets into the certain point, they evaporate. That form of the gas also control the pathogen. Because of these multi way of the mode of action, for pathogen to produce resistance against of it is very odd and not possible. It hasn't been seen or known so far. However, there are a few disadvantages of the sulfur. One of them is that it can wash off very easily after the rain. It cannot stay for a long time. So it is not effective in the cool weather, because I explained you that for the second mode of the action requires a little bit evaporation. That evaporation requires also a little bit warmer the weather. And it may burn the vines when the temperature is higher than the 90. So especially in California condition, we cannot predict those times. You gotta be keeping an eye on the weather when the temperature is gonna be higher than 87, 89, 90. You are getting into the risky point. So I'm sure you have heard about the late Dr. Gubler, who has been working on the powdery mildew diseases for a long time. And he developed kind of like the field fungicide screening trials every year that has done for many years. And when I was postdoc in his laboratory for many years ago, I have worked on these trials. Now my lab is running these trials to identify each year what combination of the fungicide is working. So every year on our campus, we set up these field fungicide screening trials. It is a randomized complete block design with the 5 replication. We use the 2 adjacent vine as a 1 plot, and then we replicate that one for 5 times. And early in the season, we use the 50 gallon per acre as you do the same thing. And then later on, we increase the amount to the 100 and finally get into the 150 gallon per the acre. We use still 430 mist blower, which is also known as the sprayer that could be mimicking your air blast. The efficacy is very important, as you know. So we know that if you have the magic bullet of the chemical in your hand, if you do not have enough coverage and efficacy, that fungicide or biocontrol agent is not gonna work. That's very important part. Anyway, so this is our trial map, which is complex. You don't need to know more about it. And these are the 5 replication in the vineyard. We use the Chenin Blanc variety, which is very susceptible for both powdery mildew and botrytis on the campus. Every year we monitor the weather. We have a weather station where we trial site. We record temperature, minimum, maximum, and also the relative humidity, and also rains, as you can see these bars. So this is the last year data, which is 2003. This year that hasn't been done yet, as this is the growing season. As you can see, we had like the very moist, unusual rains, especially close to during the harvest. But again, for us, very important, this is the other time that we get a good amount of the rain. This is the Gubler risk index. So this index is giving us basically what is the risk of having powdery mildew in your area. So now UC IPM is providing this by locally. When Dr. Gubler come up with this idea, he applied for the entire California. As you all know by now that this is not applicable because each growing season in California has a microclimate that would indicate. If you have a vineyard and if you think that you have a microclimate, you got to find out the way that have this index area. So how does this index work is that this is basically shows that the percentage of the index, as you can see, anything below these blue dots are low risk, that you have to be careful, and you can extend the intervals for the application and then continue monitoring. When the risk gets into the over these red lines, that means you have a high risk. Anytime all the conditions for the pathogen is perfect, so you have to be careful, increase your intervals. If you have been doing the every 2 weeks, you got to start doing the weekly application, depend on the label and also product. Okay, so this will give you an idea how to get ready, what to apply and what program to get into your vineyard. It will also help you to reduce the use of the pesticide in the vineyard. So these are the data that are available online, which I'm going to show you at the end of the talk, how to reach these data. So this is just the example of the data that I'm just going to explain you how to read this data. So this data comes with every year we do more than 50 different combination of the treatments. In here, you can see a program with the synthetic. All these are synthetic. And here you will see the incidence, percentage, and severity. What it means is that the incidence is when you look at the one cluster, how many berries in that cluster are showing symptom. That's what gives us the percentage of incidence. And also we calculate the severity of it, which is showing us the amount of the cluster that is infested. On powdery mildew, severity is more important than the incidence at this point, but for the controlling, they both are important. Okay, so that means that the lower number of these incidence and severities, the better. That means that product or combination of that program is working better than the other. In each trial, you will see the blue line right here. It says the pictures. When you click on these numbers, you will also see the pictures of the trial for each treatment. That will also give you an idea. So we also have the trial 2, which is the combination of the organic plant extract and biocontrol agents all together. And if you are organic, or if you would like to alternate with the organic material, so you have also those options. So these are all information that you can find. All these information I'm going to show you at the end of my talk where you can find it. This year, downy mildew was a big problem. I'm sure especially in your area, because we have a little bit more rain, which is the perfect condition for these downy mildew, or the pathogen, which is also known as a Plasmapora viticola, which is another obligate parasite of the grapevine. And then these at the beginning, shows similar symptom that the powdery mildew that could be confused by. Again, this is the back of the leaf. Again, the structure, but the most dangerous part of this disease is that it can affect the young shoots like that, and also it can completely prevent the development of the cluster. So this is a very aggressive pathogen, and if you are having this kind of symptoms, please let us know through your local farm advisors, so they can collect the samples and send it to us. We will be happy to identify if it is downy mildew or not for you. So in this part of my talk, I'm gonna give you a little bit more information about the bunch rot and summer rot, and then at the end, I'm gonna show you where you can find those reports. Bunch rot, and also known as a gray mold, is caused by the pathogen Botrytis cinerea. So these are how the pathogen grows. These are the symptoms on the berries. It is very ubiquitous fungi, which means it can colonize and attack any living organism if they find some little bit moisture, and also food and sugar especially. They love it. They colonize everything. I'm sure you know that any fruit that is the harvested, that could be the problem with this. So this pathogen attacked on grapevine fruit, especially when the Brix level is more than 88, more than 8, not 88. So which means that they like the sugar. They consume sugar better than any other the organisms. So beside the botrytis, the bunch rot, we also call it summer bunch rot and sour rot, which is mostly a problem for the table grape growers. That means that the summer bunch rot is also involved with Botrytis cinerea, but also it's involved with the Aspergillus species, more than one Aspergillus species, and also other yeast, the acidic bacteria and other organisms can be also involved. These are the very common symptoms. I gotta tell you that in the wine grape growing areas, we have seen in these kind of symptom showing before the harvest. Again, these are the table grapes. We had a lot of loss because of the late rain that we had in August 20th, I believe. We had the 2-inch rain in Central Valley, which was very, very unusual. So the one on the right, this is the Riesling cultivar, which is wine cultivar. You can see the Aspergillus colonization on these clusters. So these are the other common symptoms. It also causes the slip second, which when the yeast and bacteria get involved with the help of fruit fly, Drosophila, in the vineyard, if it is not controlled. These are some of those yeast. And then that other, the opportunistic fungi could also colonize when the berry has this kind of exposed tissue, because most of these fungi like the exposed, injured tissue they colonize. At this point, it's very difficult to which fungi are the primary or others are the secondary. It's very difficult to find out. These are the non-summer or botrytis symptoms. The one on the left is the black measles caused by the group of the ESCA pathogen that are sometimes confused by the botrytis summer bunch rot. These are the ESCA symptom on the leaves, as you can see. The one on the right is the sunburn. Again, sunburn could also be the entry point for the pathogen because you are causing a lot of determination here. Some of these sunburns also could cause by the over leaf removal to expose these cluster too much to the sunlight as well. So this we are going to talk about a little bit as well. These are the common pathogens that are involved with the summer bunch rot and sour rot. I don't want to go each one of them, but there are a lot of Aspergillus, Cladosporum, even penicillium can get into the sour rot complex. So this is the simple life cycle of the botrytis bunch rot. I'm not going to go through. That means that the pathogen can survive during the winter on the leftover dead fruits. If these fruits are not removed or buried in the soil, this could be the starting point or inoculum of the pathogen when you have the start spring flowering. Certain amount of the infection happens during the flowering because botrytis are known to act like a pollen and then get into the pollen tube and then stay there until the right condition come, because which we call it latent infection. But that spring flowering infection rate is very low. Most of the infestation comes when the Brix level start getting higher than 8, and then as you can see, the circle continues in that area. So because of this spring and also later the cluster infection, we do the multiple application of the prevention of these pathogens. So here's the how the pollen tubes gets in. It's because the spores are very small, like a pollen, and then they can just stay there, wait. So Drosophila is the one of the major insect that can distribute or help moving those fungal spores around in the vineyard. So this is how infection happens. Let's say this is the single spores on the cluster, okay? And then similar to the powdery mildew, like a seed, they also germinate. That also produce aprosorium, which is the punch point of the mycelium on the surface. And the difference between botrytis and powdery mildew is that the botrytis can melt cell wall, as you can see here, and then penetrate into the tissue of the berries, and it can't completely destroy. That's why you can have a soft tissue in that area. And they produce asexual fruiting bodies, which we call it a conidia in here. Okay. Remember, in powdery mildew, this circle happens within a week, 7 to 8 days, right? So for botrytis, this circle happens within 24 to 48 hours, 1 day to 2 days. It's so fast that's happening. So because of that, preventative the control of these pathogens are key to manage in your. So what are the factors affecting? As you remember that we talk about the disease triangle, again, even though you have a host and pathogen, the environmental condition, which could be weather, moisture, could be the good point for the pathogen to colonize, which is very, very important. Site is very important. Leftover, the last year fruits could be the inoculum source, and then unless you remove them during the dormant pruning time. And microclimate within the canopy architecture is very important. If you have a closed canopy, you have more humidity, moisture, microclimate, you will have the more good condition for the pathogen to flourish. And growth stage is very important, as I mentioned you, that they start having the risk starting from the flowering time. And another most important part of the structure of the cluster depends on the cultivar. If this is the Riesling cultivar, which when they form the structure of the cluster, that you can see some of them are so squeeze each other and then they burst. When the burst happen, sugar is just exposed outside, pathogen is in the air, they can just get in, colonize, and then it's just get the that part of the area. So that's very, very important. And also certain cultivars are more susceptible than the others. Again, so these are the number of the cultivars that are more susceptible. If you are growing these cultivars, you gotta be a little bit more careful, especially this year, because as you all know that this year is the El Nino year. We are expecting more rainy season, even though sometime in California, because of the climate change, these don't come true, but this is the expectation comparing with the last year's this year's season that we had a lot of rains. Here's the culture practices. Again, removal of those environmental conditions is very, very important. Leaf removal can help you the reduction of the fungicide used almost 50%. So what it does that when you have the leaf removal, you are having more air circulation within the canopy that reduces the right condition for the pathogen to develop. Decrease of the fertilize, as you all know that when you put the more nitrogen in your plant grows faster. Those cell walls are gonna form weaker, and then plants are gonna be, or clusters are gonna be more vulnerable, easy for pathogen to colonize. It's very important to have a balanced fertilizer and then time of the fertilizer. Again, a cluster architecture is another good point for the pathogen. So in order to have the fungicide efficacy, timing is important. We usually apply bloom, pre-close, veraison, and pre-harvest. These are the time point that you may want to target if you have one of those susceptible cultivars in the microclimate. Again, as I mentioned you earlier, full coverage of the both sides is very, very important. Again, it doesn't matter how good pesticide you have. If you don't have a good coverage, you are wasting your money and chemical and time on that one. Again, phenology is also very, very important. When you start having the Brix level more than 8, you are going to have more risk. And then if the right conditions are present in your vineyard, you got to be careful how to manage them. Again, we also do the field fungicide efficacy trial for the Botrytis, Botrytis, Samara, Botrytis as well. So this trial is taking place in a commercial vineyard in Clarksburg on a Riesling cultivar, which is very susceptible cultivar. So we set up the experiment exactly similar way that I explained you on the powdery mildew. We use in this one, we use the 3 adjacent vines as a one plot, and we replicate 5 times to reduce the error mistake in the field, because you are doing the experiment in the field. There are a lot of other environmental conditions may affect the outcome of your experiment. And then we apply, again, start from the bloom. In this year, we started in May, 100 gallon, and then later on ended up 150 gallon. And we use the same backpack to get the air blast efficacy in each site. Again, this is our map that you can also find these results in the reports. And we also look at the same climatic data to see if the conditions are good for the pathogen. Again, in August 20, sorry. So we get the good amount of the rain in entire California, I believe. And then that would increase our risk if you haven't been harvested your fruit at that point as a wine grape growers, of course. So here's the results. Again, incidence and severity that you can find those results. Each one is going to show you the individual or combination of each treatments on both synthetic. We group them as a trial 1, and also biological, organic, and plant extract. They are also known as a trial 2 that you can find from our website. Okay, here's the time. So how can you find those results? This is my lab website. If you Google my lab name, last name, and you will get this website right here. So in the top of the left corner, you will have the fruit crop fungicide efficacy trial. So when you click on that, you are gonna come to this page. Each year, it will show you what kind of trial that we have done on grapevine, and then those are the results. So when you click each one of them, you will have the results report. That report is gonna give you entire how we set up the dish trial that I explained you all the details. Everything is there, and then also the pesticide that we have used combination, and then which ones work perfect. Here's the point. As if you are pest control advisor and grower, and if you have been using the same product, it could be synthetic, it could be biological for last 5 to 10 years, you are behind the game, because every year, each companies are producing or coming with a different products, and each product has a different mode of action. In this trial, we do the trial in the field to give you an idea what products or what combination could be the alternative application for you, and then you can apply these things. So again, at the end of each year, in July 31st, we have a field trial, the field day, sorry, in these field trials to invite, you are all welcome to come join us, and then to see this trial with your own eyes, and then have together, and then have a chance to meet with the other advisors, company representatives, and also other growers. So you are all welcome, and then we provide a free lunch if that will be the driving point for you. So you are welcome to come. Anyway, so here's another point I would like to point out. So these are the pictures. If you cannot make those field trials in person, when you click on each picture, and you are gonna see pictures of each trial, 5 replication. These are each plot, and then you will see the closed caption of each treatment that will give you not only getting the results with the numbers, but also visually how they were worked for both powdery mildew and botrytis. That will give you an idea. So these are the all can be found in the website. Okay. And Cindy, how much time do I have? Do I have time?
Yes, you have 15 more minutes.
15 more minutes. Okay. So if I have 15 more minutes, I would like to give you a little bit more information about the Aspergillus vine canker, which is a part of the sour rot complex that we have been identifying the group of number of the pathogen. I would like to a little bit give you more information about that. Okay. A couple of years ago, one of our farm advisor in Fresno County gave a call us, and then he mentioned that they have been seeing these kind of early fall symptoms in one of the vineyard in Fresno. The cultivar was Grenache on freedom rootstock. The reason that we got involved with this kind of the research is that if you know that my lab has been working on the southern vine collapse, which is the combination of the viral leaf roll tree and vitiviruses, and on the freedom rootstock, it caused a collapse. That's the southern vine collapse, which I'm not going to talk about it. But the concern was if this could be the southern vine collapse, because there's a freedom and then plant are showing these kinds of symptoms. When I first came to this vineyard, first question came in my mind, as I'm sure you are thinking the same thing. Oh, this could be the viral diseases. I mean, leaf roll tree viruses showed this kind of early symptom of the fall, which was not. We screened these vineyards or grapevine. Those are showing symptomatic and non-symptomatic. They were virus free. Okay, so that's why we did a little bit more investigation about what's the causing of these early fall. So it's another picture of the that early fall symptom. So when we were examining these grapevine on the trunk, so this is the graft union right here. Above the graft union, we start noticing some kind of like the black fungal colonization. Okay, and then when you look at the close-up of these colonization, you can see that those are the conidia or conidiophore of the aspergillus. Okay, and then when we peel the bark, look at the cambium of the xylem, you can see like that this dark, wavy, black to purple color of the discoloration. That's very interesting that how the pathogen moves in the cambium in that one. So I'm sure you all are aware of the grapevine trunk disease pathogen, which are Eutypa dieback, young vine decline, Botrytis variegae canker. So I gave talk on that one in this last year's seminar. If you remember that these pathogens can infect grapevine through the wound that caused by us during dormant pruning, right? So that's why we have the pruning wound protection on that one. This pathogen seems to be penetrating in the trunk, not from the wound, that we think that we have the evidence I'm going to show you, can penetrate without any wound into the plant. Okay, so when we look at the cross section of the these symptomatic plant, we observed two kinds of symptoms. One of them is canker, which is when the pathogen start colonizing, it kills the tissue that causes the canker tissue. As you can see, look at that here. This is the phloem under the bark. In here, phloem is already gone. And then it looks like the pathogen starts getting in from here and then moving inside. Okay. The picture on the right, however, shows this is the xylem, which is the active xylem of the plant, right? And then it looks like it's also moving up and down in the xylem vessels, which is a very characteristic for the wilt. If you have heard about those wilt pathogen, they can move within the tissue. So that is showing us that this aspergillus species could be causing canker, penetrating into the plant, and also could be moving systemically within the plant up and down. So we have been working on this the pathogen for a while. We know that this pathogen has been also found long time ago in California by Themis Michaelides. That's the name where the name first identified on red globe. But we start seeing these pathogens on more northern grape growing areas and more in the wine grape growing areas. We think that because these aspergillus group of the pathogen are more adaptable to the warmer temperature and they like the hot weather, because of the climate change, we have been having more warmer and hot days. We think that these are becoming more prevalent in these other grape growing areas. In during the season, we also look at the spore load in the air. This is the simple spore trapping device that we have been using for more than 20 years. We come up with this idea for a long time. When I was postdoc in Gobler Lab, it's working perfectly. And then these are the spores. What we do is that we put these spore traps in the vineyard, and then once a week or once every 2 weeks, we collect them. These are the simple microscope slide where it is covered by the petroleum jelly, which is Vaseline, and then it's sticky right there. And then when you keep them on the vineyard, on the cordon, if there is a spores, that spores can be stick on those Vaseline. We collect them, bring them into the laboratory, and then we wash them and then culture them on the media. Then whatever grows, we count them. Okay, so in this trial that we have been running for the last 2 years, we identified that there are a lot of aspergillus and also botrytis colonies floating during the growing season, especially when the season comes close to the harvest time. Okay, so that's like the part the epidemiology of the pathogen. We identify three different the Aspergillus species: Aspergillus niger, Carbonarius, and tubangensis. So tubangensis seems to be new to California. That hasn't been found on grapevine. We still don't know whether this tubangensis has been around in California, but now we have access to those molecular identification techniques that we are able to separate those, distinguish among those species. Or these are like the new introduced pathogen. We don't know that one yet. But what we know so far is that those Aspergillus species that has been found from the vine canker, and also found from the sour rot, earlier I talked to you, and from the spore traps, seems to be similar or same species in each one of them. So we have done the pathogenicity test. This is the pathogenicity test that is showing you that sorry about that, isolates from wood, sour rot isolates, and the spore trapping showed a little bit different canker on the healthy plant when we inoculate them in the control condition. This is the control. As you can see, this is the canker. The spore trap isolates, even though they are the same pathogen, seems to be more aggressive than the sour rot and also vine canker. It's very, very interesting. We think that there's a reason for that, because those spore traps are the kind of seed of the pathogen. They are supposed to be more aggressive and then adapt to better condition than the other mycelium that we have been finding. So we inoculate them on both wood and also berries, with and without the making wound. Guess what? The pathogen was able to colonize or penetrate into the tissue without the wound, which is the most dangerous part of this kind of pathogen. Okay. And these are the results from the pathogenicity test, and they all show that they are aggressive on both tissues. And so these are the conclusions that basically what it says that we have three different Aspergillus species, and they can be in the pathogen in all of them. We have used the one calmodulin gene, which is able to separate these pathogens among each other, and then we have more the way to do that. So for you to take home message is that if you are seeing these kind of early fall symptoms, I would suggest you to have a viral panel test every year in your vineyard to have an idea whether you have a virus or not. Because as Kent mentioned you that those mealybugs could be the vector for the especially leaf roll tree viruses, grapevine vitiviruses, and then also could be getting this information. So once you have the early fall season, and if you don't have a virus, you have to look for what other pathogen could cause those kind of color pictures. Again, not every the early fall color could caused by the Aspergillus. Again, there are a lot of viral disease out there. You have to know if those grapevines are previously infected by the virus or not before you get into that conclusion. And if you have that kind of things, we will be happy to help you. So this part of the study has just published. You can access this publication full paper. If you cannot, because these the magazines are making money, they want you to have the subscription. So if you are interested in having this publication, send me an email. I will be happy to send you the copy of it. I gotta tell you, this scientific publication could be the cure for insomnia too. Okay, so I would suggest you, if you are having hard time to fall into sleep, just 10 or 15 minutes before you go to bed, you start reading these scientific papers, and you are gonna be having a good night's sleep. So these are my lab crew. I would like to acknowledge them and then take to the funding sources. If you have any questions, I will be happy to answer your questions.
Thank you, Akif. Matt is going to ask you some questions that are in the Q&A.
Of course.
So the first question we have here is, at what temperatures should sulfur dust be avoided due to poor fuming activity?
Well, if the high fuming activity is over 87, 90, you don't want to apply that. But when the temperature is lower than 50, you will not have the fuming activity, but you will still have the contact activity. So in early season, which is very common practice, our growers can start with the dusting, okay, and then in some cases they could edible. That's fine. If I cannot make a recommendation apparently as a UC employee, but if this is my vineyard, that's how I would manage it. I will continue doing the application of the sulfur until the right condition is present there. When the temperature get increased, I will turn into the program that I show you where you can find it.
Okay, so the next one here is if organic eradicant materials like Cali green are sprayed with biological fungicides, does that affect the efficacy of those materials?
No, well, you gotta read the label first. Each label of each biological chemical, they will tell you if they are compatible with biological. Okay, so don't trust your neighbor or someone tells you that yes, you can or you cannot. Reading the label is very important. I cannot tell you anything about that, but yes, there are a lot of products can be applicable together. For example, but there are also risks. I will tell you, if you mix sulfur with the surfactant oil-based products, you can have the high risk of the burning, again, phytotoxicity. You gotta read your label. Okay.
Okay, so the next one, what type of media was used to culture the sour rot shown in that slide?
Oh, we use the acidified potato dextrose agar, which is a media made of the potato, and it has a lot of sugar and also carbohydrate for pathogen to grow very fast. And acidification is the one that could eliminate the bacterial growth or culture. So that's what we usually do. We don't have to use, you don't have to use like specific media, because if you are trying to trap the botrytis, don't worry, botrytis is going to show up in your plate very short time.
Can a healthy grapevine balance nutrients have a higher immune system that can combat either PM or botrytis and reduce the need for synthetic fungicide applications?
Of course, defense. Every plant or pot has a defense mechanism. If your plant is healthier, it may fight better. Again, as I mentioned you that the fertilization is very important. If you fertilize too much, plant is going to grow too fast and it's going to be weaker for the pathogen and everything. So you got to be careful. But these pathogens are, especially powdery mildew is specific to grapevine, this species, and your defense mechanism is not gonna help that much once you have the pathogen in the right condition. Okay, you have to come up with the preventative. Of course, it's like us. So if you are healthy, you can recover the disease quicker. Doesn't mean that you are not gonna get anything. So there's always risk of. Yeah, that helps. Mm-hmm.
So next one is, what is Cober and Sapinil fungicide in the organic PM 2023 trial?
Oh, that's very specific. Send me email about that, and I will give you the more information. There are more than 60 treatments in that trial. I don't have in my all numbers in my top of my head. Just send me email. I will send you respond to email too. Okay.
Okay, and then I think just the last one here is, may you show the Aspergillus red leaf symptoms again?
Show them. Say it again. What is the read again?
It's just asking to show the Aspergillus red leaf symptoms again. I don't know if we have to share the screen again.
Okay. I don't have to show it. Just send me email. I'm going to send you the article that has the color pictures. You are going to have it. And you just Google my name and aspergillus. I'm sure some of my talk is in online, but I will send you the copy of the paper.
Is there time for more, Cindy, or?
Yes, you can go one more.
Okay, so it looks like there's one more. Or two. Well, so are there biological fungicides that can be used as standalone products that can effectively control powdery mildew?
What's the last part? I couldn't understand. It can be applied.
Yeah. Are there biological fungicides that can be used as standalone products that can effectively control powdery mildew?
Very good question. Okay. Depending on the disease pressure. Okay. So biological rule of thumb, biological works perfectly when you alternate them with the synthetics. Okay. And then the question comes in your mind, if they only work at a good amount with the synthetic, why should I use the biological? Why don't I go to the synthetics? The biological gives you an alternative, sustainable way. The pathogen has less chance to develop the resistance. The reason that we are giving you these program alternatives is that you are trying to reduce the risk of the resistance against these pathogens. That's why you need to apply those. So, for example, if the disease pressure is low, standalone biological may work at the beginning of the season. If the risk is high, that is going to give you the trigger point that, hey, risk is too high, biological may not be standalone work, alternate with the synthetics. At that time, you are going to get the synthetics at the end. So all these field fungicide trials give you an idea how to change, how to modify your spray program so you can reduce your use of the chemical, synthetic, plus how you can reduce your risk of the resistance. As you all know that today we have a resistance against the strobilurin group of the fungicide. And then I'm sure you have heard and then you have seen those. All are these programs and then application is to reduce that risk. Okay, so check those reports. There are standalone programs, combination of the biologicals and alternate with the biological with the synthetics, you will have all those information. Okay, so you have a homework. So if you have a question, just send me an email. I will be happy to respond to you.